Okay, so welcome everyone. Before I get started on the introduction, she is not going to be happy with me, but today is Angela's birthday. Happy birthday to you both. Just to lighten things up a little. Okay, so it is my honor and privilege to introduce today Dr. Carrie Breton. Dr. Breton is a professor of population and public health science at the USC Keck School of Medicine. She's also director of the Maternal and Developmental Risks from Environmental and Social Stressors Center for Environmental Health Disparities, MADRES. Her exemplary research investigates the interacting pathways of environmental health, social stressors, and epigenetic mechanisms to child health, specifically cardiometabolic and respiratory conditions. From my perspective, her work represents both the cells to society and the interdisciplinary dimensions of public health. Dr. Breton has conducted both epigenome-wide studies and targeted epigenomic investigations, a hugely complicated science from my point of view, with so many unknowns, to understand the roles of DNA methylation and microRNA underlying environmental health effects. She is author of over 170 peer-reviewed articles and PI and co-PI on multiple NIH grants. Dr. Breton holds a Doctor of Science degree in epidemiology from the Harvard School of Public Health and an MPH from UCLA. Welcome, Dr. Breton.
Thank you. So it is a great pleasure to be here today. And so thank you so much for having me. So for the next hour or so, what I'd like to do is share with you some of the work that I've been doing in the context of the MADRES Center. And today I'm going to focus specifically on ways that we are looking at the interactions and the interplay between air pollution exposure and maternal stressors on both maternal and child health outcomes, and then also on biological outcomes. Okay, I was working a minute ago. So I'm going to show you some results specifically addressing three questions. How do air pollutants and stress affect infant growth? How do air pollutants and stress affect pregnancy health? And then what are the biological effects of air pollutants? And give you really just one tiny sliver of how one might look at that question. But I want to start with some framework and why I'm interested in looking at health effects of air pollution. We already know a lot about the negative health effects of air pollutants broadly. We know that there are tons of short-term effects. There are long-term health effects on multiple organ systems. We also know that there are groups and populations who are more adversely affected or more vulnerable to the negative health effects of air pollution. And these include people with chronic lung conditions, seniors, children, pregnant women, as well as people who exercise a lot outdoors. So today I'm going to focus specifically on thinking about pregnant women and children. Why am I interested in metabolic health? Particularly when thinking about in utero exposures, pregnancy is a period of very dynamic biological and hormonal fluctuations. A lot is going on during pregnancy, which also means there's a lot of room for those biological mechanisms to be tweaked, to be affected by the exposures that pregnant women are exposed to, whether they breathe them in, absorb them through their skin, or ingest contaminants. So we also know that 40% of Hispanic women of childbearing age are obese entering their pregnancy, and 51% gain more weight in pregnancy than is recommended. And so this means that there's already heightened risk for potential metabolic disorders and metabolic complications during the pregnancy and even into the postpartum period. On the right-hand side, is also data from LA County WIC program going back 20 years now that shows clear health disparities in the obesity rates in children, particularly higher rates of obesity starting as young as 4 years of age among Hispanic children in Los Angeles. So knowing that as a baseline, when we started the MADRES Center 10 years ago, our focus was on our local community in Los Angeles, which is predominantly Hispanic population. The prenatal clinics that we partnered with recruited from catchment areas that were predominantly low-income women of color. So this image is a relatively new development we worked on over the last year to try and really represent and embody a lot of what the center itself aims to do. And so because this is an NIH-funded center, an Environmental Health Disparities Center, it does have core components of a community engagement core and an investigator development core. So there's infrastructure to support activities through those venues, but also front and center is a research and science initiative really focused on, again, the pollution sort of writ broadly and stressors, all focused in that looking at pregnancy, at the pregnancy period as a really vulnerable window of time. So I'm going to tell you a little bit more about the cohort now that we've recruited. So we recruited a pregnancy cohort as early in pregnancy as possible. We started in 2015. We ended recruitment in 2023. So in total, we recruited about 1,100 mother-child pairs. And again, we did this by partnering with primarily 2 to 3 community health centers that were dedicated to medically underserved populations in sort of the south and eastern sections of Los Angeles. But really, our catchment area ends up being pretty broad. So if you're familiar with Los Angeles at all, it's a large metropolis, and people drive lots of distances. And so you'll see a map later where the residential locations of our participants, in fact, are pretty scattered across the LA County wider geographic area. And on the right, I've just listed some of the initial exclusion criteria, which were a part of our recruitment protocol. We did try to really recruit women as early in pregnancy as possible. We ended up relaxing that criteria to a certain degree to be more flexible in our recruitment strategies. However, we did exclude participants less than 18 years of age who had HIV positive status or for various reasons could not give consent to participate. As well as currently being incarcerated because one of our, we recruited from the LA General Medical Center, which does actually treat and deliver patients who are incarcerated, as well as multiple gestation. So those were our exclusion criteria. And just to tell you a little bit about our cohort characteristics, 75% of our population self-identifies as Hispanic, 10% as Black, 8% as white, non-Hispanic, and then very few other sort of mixed groups are represented here. This is also a largely low-income population. Income data is not great in this population. A lot of women say they really truly don't know what their actual annual income is, but when we do get concrete numbers, the reality is that more than half of the women have an annual household income of less than $50,000. And in Los Angeles, that certainly does not go very far. It's also a fairly lowly educated population with more than 50% of the population having a high school education or less. And about half were employed and half were unemployed at the time that they were recruited into the study. So I told you I'd give you a little bit of a map of LA and see if show you that sort of this map is sort of the greater Los Angeles area. The black dots are all of the participant residences, and then outlined in pink are how we defined our MADRES neighborhoods based on more than 5 people living in a particular geographic boundary. So then we can look at the MADRES neighborhoods outlined in pink and overlay that over all sorts of other GIS-based information that we might want to look at. So shown in the middle is just the neighborhoods overlaid on census data for race and ethnicity, where purple are predominantly Asian neighborhoods, green predominantly Hispanic neighborhoods, and yellow are predominantly Black neighborhoods. So I think what you can see is, again, a lot of our neighborhoods overlap with communities that are predominantly Hispanic. And then on the right is actually redlining information in Los Angeles. So the neighborhoods in yellow and in red are historically redlined neighborhoods and communities in Los Angeles. And so again, what you can kind of see, I hope, is that a decent number of our of our MADRES neighborhoods, in fact, are in communities with have historically been redlined neighborhoods. So one additional way that we were interested in trying to understand an overall assessment of neighborhood burden, and I'm going to share with you sort of this index now because it'll come up again in some of the analyses. So we use the Cal Enviroscreen 4.0 index to try and get a sense or ranking of communities across the entire state of California for their sort of cumulative impact or environmental burden. So there's an overall score shown on the left, and then that's really just a multiplier of both two subscores. One's an pollution burden score, which is really summarizing characteristics like how close to a hazardous waste site do you live? How much air pollution are you exposed to? And then there's the population susceptibility subscore that sort of accumulates data about asthma rates, about low birth weights at the neighborhood level. So what you can see from this map, blue is are the low scores, and pink and red are really high scores on this overall index, which ranges from 0 to 100, is that a lot of our communities rank pretty high on this CES score, and also if they're high on both the population susceptibility subscore and on the pollution burden subscore. So we can look at the overall impact, and we can also break it down and look at the subscores if we want. And some of the work that we've been trying to do is to use this index as one indicator of potential vulnerability when we're looking at then individually measured exposures or other individually measured psychosocial stressors, and try to understand whether there are any differences in disease risk. So the first question is, how do air pollutants and stress affect infant health? I'm going to share with you some data now looking at fetal growth, at birth weight, and then in growth during infancy. And then in the moms, we'll talk about some pregnancy complications like gestational diabetes. So we measure air pollution a couple of different ways. We have ambient air pollution measurements for NO2, ozone, PM2.5, and PM10. This is based on inverse distance weighted modeling approaches. Then we also have traffic-related NOx estimates from a K-line 4 line dispersion model. This was in partnership with Sonoma Technology, Inc. We have a lot of meteorology data, and then we have some newer ensemble-based machine learning models for weekly NO2 and weekly PM2.5. And these are newer models that my colleague Rima Haber has really been at the forefront of developing. One way that we captured stress in pregnancy was to ask the perceived stress scale at each time in pregnancy. And so this is a 10-item well-validated assessment of stress that has been used for many, many years. So we have looked at, and we actually measure this in pregnancy as well as postpartum, so we can continue to look at these self-reported stress measures across a decent period of time now. And we started by looking at fetal growth outcomes, and this we measured these during a 3rd trimester ultrasound visit where we brought the women into our clinic space between 30 and 34 weeks. And we conducted, we had an ultrasound tech who conducted the fetal biometry. So we had head circumference, abdominal circumference, biparietal diameter, femur length, and estimated fetal weight, all the sort of standard fetal biometry that you would have measured during your OB/GYN visits as well. And this effort and analyses were led by my student, Alicia Peterson, at the time. And what she did was she used a distributed lag modeling approach to look at windows of susceptibility across pregnancy and even into the pre-conception period. So we had detailed information on their residential addresses, and we assigned air pollution exposures pre-pregnancy and then all the way through pregnancy. So we looked as far back as 12 weeks prior to conception, and that's the Okay, that's shown on the left-hand side of the blue dotted line, right? So this is, I'm looking at preconception exposures and then pregnancy exposures. And what we find are no obvious associations of preconception air pollution exposure with fetal biometry, in this case, abdominal circumference. But what we do see is that a pretty consistent effect where higher PM2.5 is associated with lower abdominal circumference in the third trimester, for windows of exposure that really range across the whole first half of pregnancy from week 2 to week 23. We see very similar results if we look at estimated fetal weight as well. So again, on the left, no evidence for preconception associations or effects of air pollution preconception. But in this window in early to mid-pregnancy from about 4 to 18 weeks of gestation, we see an inverse association. Again, higher PM2.5 exposures are associated with lower fetal weight. So we wanted to extend this and look at birth weight, and this was work that was led by postdoc Jason New. So he first looked at the 4 ambient air pollutants using the same distributed lag modeling approach, and saw, again, for PM2.5 and NO2 this time, really similar results where higher PM2.5 or higher NO2 are associated with lower birth weight, but that window of exposure is really in this mid-pregnancy period, a little bit narrower window in this case when we're looking at birth weight as the outcome. But this is actually really corroborates a lot of the data that already exists. So this is not necessarily new in and of itself that air pollution is associated with low birth weight. That's pretty well known. What is, I think, a little bit more new is that we're trying to really identify which periods in pregnancy are most vulnerable or most susceptible to the air pollutant effects, because then we also could think about, okay, well, what's going on biologically at that moment in time during those weeks during pregnancy in terms of thinking about mechanism. What's also, I think, new, and what I want to share with you is the next step that Jason took, which was to look at the effects of air pollution on birth weight in groups of women who were either highly reported high stress or low stress, or lived in highly burdened neighborhoods or low burdened neighborhoods. And so on the left is the result for PM2.5. And again, what you can see is the most negative effects on birth weight here, which was basically a decrease of 700 grams in birth weight, are really among the women who report high perceived stress and live in neighborhoods that score really high on that CES score. So they're highly burdened neighborhoods and women who are then experiencing their own reflection of their perceived stress as being very high. That's the group of women where air pollution is having the greatest effects. And we see that with PM2.5 on the left, and then NO2 on the right. NO2's pattern is a little different. So if you'll notice, there's the bars are actually the greatest effects on lower birth weight are in the group of women who reported high perceived stress, right? So it didn't really matter which neighborhood they were living in this case for NO2. So what we're finding is this interaction really between various aspects of stress and neighborhood level burden with a personal assessment of air pollution exposure on birth weight. So we wanted to extend this further. We're now following the kids, they're growing, they're getting older. So we had a lot of data on growth in the first 2 years of life. And that's the spaghetti plot shown here. And this is a combination of data points from the visits that they come in and see us in person, but we also abstracted medical record data from the pediatric medical record. And so in that first 2 years of life, babies come in, they get measured a lot. So we have a great density of data points for growth trajectories. And so that's what we did. We looked at a comparison of the 10th to the 90th percentile in air pollutant exposure and used piecewise linear spline modeling approach to really try to capture both the growth trajectories in that 2-year time period as well as the attained weight at age 2. And we could look at both outcomes in this modeling framework together. And what we found was actually somewhat convoluted. So air pollution effects had a stronger effect in the first 3 months of growth, in this period where you might consider it to be sort of an infant catch-up growth. So PM 2.5 and NO2 were both associated with steeper growth in that period of time. But then the growth rates actually flattened out, so that attained by age 2, the association with pollution and attained weight was actually negative. So we were seeing lower attained weight at age 2 with higher levels of PM10 and NO2. And this is not exactly what we hypothesized. I think a lot of evidence in the literature is more suggestive that air pollution is going to be obesogenic. We are thinking we would see higher rates of obesity in the kids. So this is something that we are still sort of trying to understand what our data are showing. One of the things we want to do is continue extend the analyses into later ages, right? Because as we've got more and more data coming in now with growth as we follow the kids up and they're getting older. So that work is ongoing. And so now I'll switch gears and talk a little bit about maternal health outcomes. And then my second question, which was similar in vein, how do air pollutants in pregnancy and how do stressors in pregnancy potentially affect maternal health in pregnancy? So thinking specifically about pregnancy complications like hypertensive disorders of pregnancy, in part because these are, hypertensive disorders in pregnancy are one of the leading causes of maternal morbidity. Gestational diabetes is really common. It affects up to almost 20% of pregnancies each year. And we definitely see rates of GDM in our population as well. And we also know that pregnancy complications lead to greater risk of developing cardiovascular disease and metabolic diseases later on in terms of the women's health post-pregnancy. I'm also quite interested in thyroid health in pregnancy. Maternal thyroxine is in that first half of pregnancy is critical for a fetus's neurological development. This is well known, but subclinical hypothyroidism is also increases the risk for maternal complications in pregnancy. And we can measure thyroid hormones and we can look at this. So I'll first share with you some of the results looking at gestational diabetes and glucose intolerance in pregnancy. We had about 12% prevalence of GDM in our population and a 23% prevalence of glucose intolerance in our population. And so both Jason and my colleague Sheree Farzan really led this work here to look at how air pollution may affect risk of GDM and GI in pregnancy. And by and large, we do not see any associations with glucose intolerance. But what we do see is an increased risk for gestational diabetes for all three pollutants that we looked at here, PM2.5, PM10, and NO2. And you can kind of see it's pretty consistent. The red dots show the sort of significantly elevated windows, right? In this window in pregnancy, that's really about 2 weeks pre-conception to 2 or 3 weeks after conception. So right around the time of pregnancy, exposures to air pollution seem to be increasing the risk of development of GDM in pregnancy. And again, we took that one step further and looked at these results stratified this time by women who lived in communities with either the high CES score or the low CES score. And here the results are a little bit more convoluted. So for PM2.5, the data seem to suggest the higher risk of GDM really is among the women living in the high communities with higher CES score, higher neighborhood burden. But we see an opposite association for PM10. So it's a little bit mixed and a little bit less clear about what's really going on here. And I think one of the things that would be nice would be to look at other indicators of neighborhood characteristics. And that's not something we've done yet. We certainly have the capability of doing that, but it just hasn't happened yet. In terms of maternal thyroid, we measured thyroid stimulating hormone and free thyroxine, as well as TPO antibody in pregnancy in the first trimester, which is really when some of the neurological effects and influences is believed to be greatest. And then, the good thing is that we actually had pretty low rates of subclinical and overt thyroid disease. So we did look at these categories and we did try to assess that. In general, our rates are too low to really look at from a modeling perspective. So we just looked at continuous values of these hormones in the analyses. And what we found, again, were some associations for PM2.5 and PM10, where both of those pollutants were associated with higher TSH levels in pregnancy, specifically for exposures in a window of exposure between 3 and 5 weeks of gestation. Now, these DLMs are not spanning all of pregnancy, right? This is a much narrower, truncated analysis because our measurements of thyroxine were only in the first trimester. So they're only happening in the 12 to 14 weeks. So given some of the restrictions with DLM modeling approach and the nature of when we were looking at exposure and when we were measuring thyroxine, we were only really looking at preconception and very early pregnancy as risk windows anyway. But again, we were seeing some association, some evidence for pollution affecting TSH levels, no evidence, no associations with free T4. And then this time we actually looked at two indicators of neighborhood deprivation, wanting to understand whether the air pollution exposures might affect thyroid levels differentially based on we used the Gini index of income inequality, as well as the area deprivation index. And we just dichotomized these scores at the median for the purposes of looking at high and low categories. But what we find, if you look for free T4, we didn't really see anything overall, and we didn't see anything stratified. But what we did find for TSH, particularly where we had effects for PM10 and PM2.5, are that the effects are strongest, the blue bars, the effects are strongest for women who are living in neighborhoods with higher income inequality or that had higher area deprivation index. So again, we're seeing some differential effects on how air pollution may be affecting thyroid health based on some of these neighborhood characteristics. So just to summarize the prenatal effects, we found with air pollution that PM2.5 in particular is associated with reduced fetal growth, fetal weight, and birth weight, and that some of these effects are stronger in individuals experiencing high levels of stress and living in more highly burdened communities. We see similar effects for increased risk of gestational diabetes with PM and NO2 in this case. Again, also some of these effects vary by neighborhood burden and air pollutant effects on thyroid health as well, specifically with looking at TSH levels, where the effects are stronger in some more highly burdened neighborhoods. So now I'm going to switch gears a little bit to ask the question about how biology might be responding to air pollution. And this is interesting to me from a developmental origins hypothesis point of view. A lot of potential future disease risk in the child could originate in the in utero environment. And it has shown you a lot of potential effects on child growth with air pollution exposures in particular. But now the question is kind of, okay, can we figure out in utero what biologically might be changing or happening in response to air pollution exposures? And this is work that's really being driven by my postdoc, Helen Foley. And so what I'll share with you today is some work looking specifically at microRNA. And actually specifically microRNA that may be packaged in exosomes or extracellular vesicles. So these are small packages, basically little biological packages. They carry lots of different things, RNA, proteins, lipids, et cetera, but they also include microRNA. And this is an attractive target to think about, particularly in pregnancy, because we know there's a lot of maternal-fetal communication. And so these exosomes and extracellular vesicles may originate in a maternal target organ, may target the fetus, and provide one biological way of communicating. We also know that there's evidence already to suggest microRNA are associated with conditions like preeclampsia, preterm birth, gestational diabetes. There are people who are interested in developing early detection biomarkers for these types of pregnancy outcomes from a clinical point of view. But we also know that pregnancy is very dynamic, including microRNA, and there's a lot of change, just normal change over pregnancy in terms of microRNA expression levels. So we want to try and understand, okay, given that context, what does air pollution, do the air pollutants that we can measure have any effect on microRNA? So to do this, we extracted extracellular vesicle microRNA from plasma that we took in the first trimester, and in the third trimester, we measured the microRNA using the nanostring platform, so that quantifies about 800 microRNA at the same time, and then we linked it to our other data. And we looked at exposure windows specifically 1 week prior and 8 weeks prior to the time that we measured the plasma and assessed the microRNA expression level. And this was really to try and get at either an acute effect or a sort of subchronic effect of pollutants still in that pregnancy period on the microRNA. So what we found was that both PM2.5 and PM10 were associated with several, about 50 or 60 microRNA. But this was only in the third trimester and only for that 8-week averaging period of time. So the sort of subchronic late pregnancy microRNA effects from both the PM2.5 and PM10. Also, what you'll notice in these volcano plots is they're skewed heavily. You see a lot more red than you see blue, right? So basically, we see a lot more upregulation in both in the microRNA targets from these air pollution exposures. So what does this mean biologically? Well, microRNA can affect a lot of downstream gene targets. And so there are pathways that you can use and programs you can use to predict the gene targets for microRNA. These are not specific to pregnancy because the databases are not finely tuned enough to really give you that. But we can predict the gene targets, and then we can feed all of those gene targets into various different pathway analyses, try to understand what biological pathways might be being triggered here. And what we do find is enrichment for some inflammatory and metabolic pathways. So this is interesting. Okay. What we also can do is look at a human microRNA disease database. So this basically is a database that already has linked certain microRNA to known diseases. And 92% of the 103 microRNA that we had identified being related to air pollution were associated with lung conditions in this database. So this makes sense. We're talking about air pollution, the lung is a primary or target organ. So, there's a little bit of, okay, we can maybe believe our results a little bit more because at least it's making sense that we might expect some things to be targeting the lung. But also interesting is that some of the microRNA that we found were also associated with preeclampsia, with preterm labor, with inflammation and oxidative stress. So pathways that are already implicated for air pollution health effects. And or relevant also to the context of pregnancy, which is what we're talking about and what we are interested in understanding. Also, adding some more confidence, if you will, into the results are the fact that some of these microRNA have also been previously associated with diesel exhaust, with wildfire smoke and tobacco smoke. So other airborne exposures that are different, but may trigger similar pathways. So just to give you kind of an example spotlight, one can dig deeper into the microRNA if you want to. So if you take MIR 187, for instance, this was one that popped up in association with pollution. This microRNA targets inflammatory TNF-alpha in lung epithelial cells. So it's going to be in that database for lung, right? It does so target via like an anti-inflammatory IL-10 mediated signaling pathway. So it's involved in inflammation, and inflammation is one key pathway that is, by which air pollution may be acting. So MIR 187 is also tied to preeclampsia. So this is kind of interesting and may suggest other research questions, frankly, to ask, which is, maybe we should really actually be looking at air pollution and preeclampsia in our population, and even potentially trying some mediation analyses with microRNA as a potential mediator. Not stuff we've done yet, but could be interesting to do. So similar to air pollution, because we've been wanting to bring air pollution and stressors together, we also then looked independently at some of the different stressors that we have assessed and whether they also could trigger microRNA associations. So I told you about the PSS already, but we have also looked at depression scale and a pregnancy-specific distress questionnaire, which I'll show you in just a minute. But the rationale for doing this as well is that, in and of itself, we know that there's a lot of stress and anxiety and prenatal depression affecting our pregnant populations, including our own population. And circulating microRNAs have been of interest in as potential biomarkers for these diseases as well. And there has been some evidence for that already. So they could potentially be good biomarkers. So the postpartum distress measure was another 10-item scale that we used and we looked at, I think, in the second trimester. And so it's developed specifically for anxiety and depression related to symptoms of pregnancy. So a little different than the PSS. So we wanted to look at that. And then we looked at the Center for Epidemiological Studies depression scale. So this is, a well-validated scale that, gives some indication of risk for potential clinical depression above a certain threshold. So we looked at that too. And we've actually measured this one at every trimester and pretty much every year thereafter. So we looked at all three of those scales in association with microRNA. And what we found, this is showing you sort of the same types of volcano plots. So we found a set, again, of about 50 microRNA that were associated with at least one of these stress measures. 17 of them were shared between 2 or more stress measures. And then 2 of them, near 150 and 148, even met sort of an FDR adjustment for statistical significance. So they were like the 2 that were probably the strongest associated with these stressor scales. So we again did some pathway analyses. We fed the microRNAs into the predicted gene targets, took those targets and fed them into pathway analysis using the Panther program. And what we found were 15 significantly enriched pathways, sort of illustrated on the right are those pathways for each of the 3 stress scales. But what's most interesting, I think, is that 7 of the 15 pathways were shared between all 3 stress measures. So even though you have different stress scales capturing different entities, what we're seem to be seeing is that some of that is biologically being funneled through the same pathways. And this, I think, is really quite interesting. So are some of the biological responses may be redundant, maybe and triggering some of the same pathways. Okay, so what we want to do now, we've looked with microRNA, we're not as far along as say with some of the health outcomes analyses. So we've looked at air pollution independently and the stressor scales independently. But what we haven't yet done is put it together. And that's one of the next steps, is to really kind of put it together, trying to understand if there are, again, shared biological pathways or completely independent biological pathways being triggered by these exposures. But in addition, we have some other kind of goals looking into the future. One of them is that this was all done in pregnancy, but we have samples and biospecimens in sort of in the postpartum period as well. And we'd like to understand whether the microRNA profiles change postpartum, and then whether either the prenatal environment or the postpartum environment from the point of view of microRNA could be predictive of any downstream cardiovascular disease risk or mental or depression risk, basically. So those are actively under investigation right now, some of that modeling and data analysis. So I'd like to end with the last couple of slides, really talking a little bit more, giving you a sense a little bit more about other scientific initiatives in the broader scope of the MADRES Center, and tell you just a few other aspects of the center and scientific directions. So I've shared with you some of the research that I've been leading, but we have a large team. I have several colleagues leading other areas of interest. So in the center and at the core, in the center, in the middle of the figure in our center, at its core is an interest in really, as I've mentioned even in the beginning, understanding a whole suite of environmental exposures, not just air pollution, but heavy metals exposures, novel chemicals, pesticides, and really how those environmental exposures can affect maternal and child health in the context of very different individual and social stressors. So we have work both on the biomarker level, looking at cardiovascular and metabolic biomarkers, allostatic load, epigenetic and aging biomarkers. I showed you some of the epigenetic work, but we have some other avenues going on as well. And then in health outcomes, we have a whole effort around brain health, both in the moms and the baby. We have more going on with pregnancy complications and cardiometabolic health in the postpartum period, particularly. And then additional child growth follow-up and looking at body composition and other measures beyond just sort of the BMI data that I showed you. So this is all ongoing work, but we are also poised for the next 5 years of the center, fingers crossed, And what that looks like actually is we've pivoted a little to more of an interventional approach. So in the future 5 years, what we hope to do is we're going to focus a little bit more on metals. We're going to go back to our metals data that we started with actually, and really look at the postpartum cardiometabolic health of moms in the long term, really relating to metals exposure in pregnancy, but also postpartum. And then have an intervention that we have planned to go into the homes of our participants who now have young children. And the goal of the intervention is to just reduce overall metals burden in the home. And so it's sort of this multi-pronged approach that's tailored to each individual family. But that's at least the grand ambition, where we would be able to see measurable reduction in metals levels over the course of I think it was like about a 6-month period. So that's the future of the science. I come back to sort of our overall picture of the center because I wanted to spend 2 slides on community engagement and training future generations. So in terms of community engagement and scientific dissemination, I think one of the core aspects of our center is really how do we both disseminate our research, but also respond to community concerns. We've done a variety of different types of things. We have a team that's actually really good at producing infographics. We have a lot of infographics that are either direct summaries of research results or tailored toward a community need. So we have some stuff on bed bugs because bed bugs were a problem in some of the communities. We have heat-related infographics because our summers are getting really hot. Wildfires, whole other story. So we have a lot of infographics. We've done policy briefs during the COVID period. We've worked with artists. The image that I started with, and that's shown here, was actually a partnership with an artist in residence at USC to have him sort of interpret what our center mission is in an artistic viewpoint. And so that was really interesting and wild for me because I've not done anything like that before, but it was fun. And then we produced some videos, isolated videos here and there, depending on the need. One of the mainstays of our community engagement is also to conduct workshops in the community on environmental topics related to needs of the community members. So we thought we would start with like an introduction to air pollution, like an air pollution 101 for community members. But at the time, they actually weren't interested in that. They're interested in it now, but they weren't interested in it in the very beginning. And so in the very beginning, it was much more tangible concerns about like cleaning products, for instance. How do we make green cleaning products? How do we make beauty products that are not toxic? So we have sort of hands-on workshops that address some of these concerns. And then we have some workshops that are more geared toward training the trainer models too, in terms of being able to maximize impact in the communities. So we do that. And then in terms of scientific dissemination also, we are, I would say, trying to get better at working with the press. Some of that doesn't necessarily come naturally to many of the people in the center, including myself. So that has been a growth area. And I will say that over the summer, we had a paper on fluoride and neurodevelopment in our cohort that kind of blew up over the summer. And so thankfully for me, I was not the last author on that paper. So I did not have to deal with the press. And I was very happy about that. But my colleague, who will visit next week, got to bear the brunt of a lot of that. So anyway, so we do our share of that as well. And then the last thing I'll just say is that one of the big missions and big effort is to train the next generation of environmental health leaders. And so we do this in 2 ways. It is a formalized part of our core, but we have an internship program. We partnered with Cal State Northridge. And so the interns all come from Cal State Northridge. They get placed in MADRES, matched with their research interests. I will tell you that most of them want to do community engagement work. Most of them have grown up in the communities, and this is where they want to go back to, and they're getting their MPH, and that's what they want to do. But we will place them as best we can, given their skill set and their interests. And then we have a pilot projects program. So we fund projects that are specifically on environmental health disparities projects. And those are geared toward postdocs and junior investigators, assistant professors at USC or CSUN or CHLA, which are our partner institutions. And so these are the faces of our trainees thus far. So we're getting a decent cohort of them now. And it's been fun to see, where they end up. We have one person who started as an intern, became a PhD student, worked with us for her PhD, and is now like a postdoc at Kaiser, trying to write her own K99. So it's great. It's great to see kind of the career trajectories get launched. I would just like to end with a series of thank yous and acknowledgements to the large team that over 10 years has made this possible, right? Not everybody's been with the center for the entire time, but everybody has contributed to making the center what it is and to conducting the science. And this is team science. There's just no way that one person can do this type of work. So that is our team. So, thank you.
I have a question also.
Sure.
So first of all, I mean, this is tremendous. I think you're going to solve all of the global warming.
No, don't say that.
I mean, you hit all the risk factors between air pollution, low birth weight. I mean, it's amazing. The problems that you're working on. So my question is very specific to your finding for TSH.
Yeah.
And I'm curious, given the association with economic conditions, if you also looked at exposure to iodine.
Yes, we did. And I almost included those in here, and I didn't in the end. So interestingly, we see there is it does differ by iodine status, but the effects are in the iodine replete group, not the iodine deficient group.
Interesting.
We are trying to publish that right around now.
Yeah.
And it hasn't quite been submitted yet. It's somewhere close.
So no, did you look at like fortified salt versus seafood?
No,
they're not eating pink Himalayan.
No. We measured. No, we have dietary data that could be investigated more that we have not done because we have diet recall data. But the problem is it's in the third trimester, and this was really first trimester. You'd have to make some assumptions a little bit, but we could play with that a little bit, but we haven't yet.
Interesting. Yeah. And iodine replete makes sense.
It was.
Makes sense.
Well, we were sort of thinking that like, if you're iodine deficient, you've already taxed your system so much, right? That that's going to be the main driver of anything, but maybe you wouldn't see the air pollution effects.
Right, then you see that.
Because they're presumably going to be, that iodine itself is going to have a much bigger impact, but I don't really know. Yeah.
Thank you. First of all, that's terrific. You showed these interactive stressors, environmental stressors, but also sort of gravel neighborhood-led stressors, presumably all sorts of other bad things going on. And you find some of which we've seen in our work, particularly on mental health, more stressors, worse, more stressors, worse. So can you actually comment a little bit about size of effect? What was, because I couldn't quite piece it together from the graphs you were showing. What's the relative size of effect that you're seeing linked to pollutants? Is there a way for someone who's not an environmental scientist to quantify for me size of that effect versus, say, size of effect of other bad things, non-environmental?
Sure, that's a good point. We probably haven't done a great job of that, but I would say, so when we use the distributed lag models, it's a little hard to extract that immediately. What you can do is sort of then create do another more conventional model looking at the weeks, just averaged week of exposure from your window of interest that you've identified. I think I think it was about, well, actually, I think I have it on the slide, but somewhere in the 18 to 20 gram type of difference on a weekly basis from the distributed lag models. But the birth weight differences was really large. It was 700 grams. Where all of that effect was really in one group of women. So the birth weight was actually, was pretty large. I think we don't have as great a Well, maybe we do. As great of benchmarks for effect size when you think about it from a week by week perspective in pregnancy. But we probably could actually do a little bit of those calculations and make a better interpretation because, there's standardized growth rates at any given, within the first trimester, within second trimester, third trimester, with most of the growth happening in that third trimester anyway. So we probably could benchmark it better.
Yeah, no, translating our work in particular.
Yeah.
We struggle with that.
Yeah, no, and it's a real struggle.
And there's a relative effect.
Yes.
In terms of telling people.
Yes.
This happens.
Yes. And we could probably very easily look at it and compare it to smoking because everyone thinks about smoking and smoking effects, right? So that would be a first pass for sure.
Thank you. I think maybe it's closer to Sandra's question just related to stressors is similar to what he was saying about our work. You're thinking, or at least from my understanding, was you're looking at perceived stressors and then deprivation of the neighborhood.
Yeah.
I was wondering if you have any interest in, and this is just very selfish to me, and looking at we're trying to ask people about economic and financial stressors at the individual level, especially because I saw that next steps you were looking at like SDOH and metal.
Yeah.
So I was wondering if you ask people if you have data on that, or if you're interested in asking more people in the future about the actual stressors they're experiencing.
So we did ask, I think it was just like some small 5-item financial stressor scale in the second trimester. We do have that data. We have not looked at it as much. I think the handful of times we haven't seen as much with it. It was just given the way we roll things out, it was in the second trimester, so it doesn't always line up with like when we did the biospecimens, which is okay. And I mean, because I think you can make some assumptions about the stability of that. But we do have, the short answer is we do have the ability to look at some element of that. And for all of these analyses, we haven't done it yet.
Thank you so much. That's fascinating. Seeing your passion for science and training the next generation is impression. Very, very much. Thank you so much. So my question is on the PM 2.5.
Yes.
No, LA is quite unique. Because PM, I was wondering if you're mapping if the emissions are the main contributor or bushfires. So those, particularly in LA, those things are more hard to identify, perhaps. And I think they are important to inform policy changes at local level, community action, for instance. So any thoughts on that?
Yeah, so we have actually a whole effort on that Rima Haber leads on source apportionment modeling for pollutant exposures and trying to understand. And actually, I think we've had a couple papers now come out. Just like, especially in PM, like what are the primary contributing fractions, and do we see similar associations with we've done some personalized monitoring on a subset of pregnant women, and how well the personalized monitoring levels predict like low birth weight relative to the ambient data that I showed. It doesn't line up perfectly. And there's not a great correlation between the personal monitoring data and the ambient data, right? But so we do have a whole line of work looking on that. Also on, I know I was going to say wildfires. So we had an administrative supplement, a climate supplement in the last year to look at heat and wildfires, and Rima's also leading some wildfire work in another project. So we can look at the wildfires across our timelines and how many times a woman has been potentially exposed to wildfire smoke. LA, none of them have been exposed to like actual fires burning in their neighborhoods where these women live, really in the center of Los Angeles. So, but it is a bit more about the smoke exposure that they may have experienced. So there's active work looking at that now, and also looking at heat exposures. And I have an R21 I put in to look at wildfire exposures and microRNA. So we'll see.
I have a question from Zoom. So from one of our Zoom.
From Zoom. Okay.
Yeah. So recognizing that moving to different neighborhoods may not be feasible for these mothers, and that policy level impacts on air pollution would take a great deal of time, what tangible recommendations do you have for pregnant individuals based on these findings?
Yeah, sadly inadequate, as I will say. Having just experienced a wildfire, I have a greater appreciation for the gaps that currently exist in terms of data and recommendations. But what we have historically done, and so I think there's twofold given this research. One is you can certainly work on stress reduction in pregnancy as a modifier, so at least at the individual level, right? And then there are behaviors you can do to improve your air quality in your home, and in your commute, and in your workplace potentially, depending on that will depend a lot more sort of on your work situation, right? So you can have air purifiers. You can even just know how to read the air quality data. The AQI is now readily accessible on your weather maps and things like that, for just baseline information on the outdoor air quality. And I mean, it's sad to say though, like in LA, the summer's always yellow, right? On the AQI, right? You're never good. You rarely have good air when you're, running around and your kids are playing sports outside. So there's a little bit of, you've chosen to live in LA. So this is sort of what, this is a part of that choice that you made, whether you realize it or not. But you're right. You can't, move away very, that's not usually a realistic, heck, that's not even a realistic assumption when you talk about like wildfires that have upended people's lives, and they're like, and now you've got even these huge effects, and you have some people out there who are like, I'm telling my kids to move to the East Coast and get the heck out of Dodge, and but that's pretty scary messaging that you really can't have the entire city of LA like leave. So yeah.
We have better air here, I think.
Tornadoes and floods.
Right.
Well, I'm going to ask you about whether LA's got so much commute and people are in so many different places. Do you try to account for workplace or other exposures, or is it all pinned to their residence?
We collected a lot of detailed information on commute and occupation. And we have not done as much with that data as we should have. I think in our population, we've looked at the commuting data a little bit. The reality of some of the participants is that they are, they're not driving in their car. You're not driving in cars where you can kind of put your air recirculate on and be all good. They're taking multiple buses. They're walking neighborhoods, and so their commutes are in environments that they don't have as much control over. But we have looked a little at commuting time and some estimation of time weighting the exposures. And we certainly have looked at mobility and moves, and we have accounted for moving locations in the air pollution exposure assessment. But then in terms of other exposures or occupational exposures, like I said, we do have some, we made an effort to like collect that data. We had a postdoc that was kind of come and focus on occupation and then didn't come. And so it just really hasn't.
So maybe we let the birthday girl have the last question.
Oh, do you have time? I don't know if it's somewhere around seasonal variation with pollutants, and if there's seasonal variation. Also, you mentioned hotter summers, the potential of higher ozone. And Southern California being non-emission zone for and if you're looking at specifically ozone.
We have looked at ozone. We just don't see much with ozone. We have looked at many different ways of adjusting for season or looking at season. Either crudely, some would say there are only two seasons in LA: warm and warmer, I guess. Warm and hot. So we've looked at that, but we've also used meteorology data. We're doing a lot more with just actual temperature and heat and stuff like that, and with the air pollution modeling. We haven't looked as much in terms of modification as we maybe could, but we do try to account for it.
Okay, so thank you very much.